Okay, Brooke. So, oh, what's going on here? All right, so our goal is to check out whether the intron losses and gains that we're seeing are real or whether something's funny. Specifically, to look at whether there are paralogs within the genome that contain the other state. So, if we have one gene that has the intron, is there also a gene, another copy in the genome that lacks the intron? Okay, so we're going to start with, this sort of overlaps the end of the last tutorial I made for you. We're going to start with the HI files, the raw HI files that look like this. And what you can see is that in the outputs, we already have some annotation of these things. So here's an intron position. As you see, it's actually conserved across all 4 species, but it does not have a good enough local alignment because of this gap nearby, this gapped position. And so it's been annotated as bad local alignment. So we have one more script that's going to work on this HI output and find positions that are phylogenetically informative, that is, positions at which introns are present in exactly 2 species. So that could help us resolve the relationship. And where they have decent local alignment, and also where they're in genes that are not intronless, genes that contain introns. Let me have a quick look. Make sure. Oops, I had changed this. Well, we'll leave it this way for now, actually. I'm including introns that fall in genes that are intronless. They seem to have the same pattern. It may give us more characters. It's hard to know. Those aren't necessarily bad. So we'll leave them in for now. Okay, so the way this works is we're going to run our script, Perl nearly intronless, on the HI file, and then we can just save that as human buffer cod Medaka positions. And so now we can have a look at that file. And what we see is this is the output of grep, which is a command line tool. And each 41 lines is a position. So these double dashes here mark, they demarcate the position. So this is one position. So that is an intron position that's informative and the flanking positions. And then if we go down further, this next batch is the next position up until the next double dash. So this is the second position. Okay, so and as we see when we look at the intron position, we have a single, within this largish region, we have a single intron position, and that intron is present in the first species, the third species, but not the second and the fourth. So that means it's present in human. It's present in human and cod, but not in puffer and or Medaka. All right, so now we want to study this local region and figure out more about what's going on. So we have this position in this kind of abstracted context where we don't know exactly. We have a little bit of local alignment, but we don't have the whole thing. So the first thing we need to do is work from this intron position to get the overall sequence. And there are a few ways we could do that. So what I think we'll do is we'll go ahead and get the local protein coding sequence from human. And so that protein sequence is DVTGGLGTDELRLLYGMAL, so and so forth. I think I don't know. There are a few different ways to do it. I think we might as well just do it by just write that down. Oh, we actually need an uppercase. There's a script called uc uppercase.perl that will give us that back in uppercase. And then we can just look within the human. So there's this file. This is the human protein. This is the exons introns files translated, and so we can just go get our sequence of interest in there. Various ways to do that. So I'm just going to search for it, and there it is. And so this is our sequence of interest. But actually, we don't want to get the whole thing. We just want to get the local. We just want to get a little bit of it, a little bit of the local sequence. Oh, so I messed something up. So we'll try it again. Okay, so we're going to grab out some of this sequence, the flanking sequence, so that we can use it as a blast search against the fish genomes to see whether they have multiple homologs of this gene, some of which do and some of which do not contain the intron. All right, so the intron is right here in this R. If we look back at the file, we can see it's in this R. So we basically want to ask whether we see an intron, whether we see breaks in the genome at that position of that R. So we're just going to have a file that has some dummy name and contains the sequence of interest. And again, let's orient ourselves here. Okay, so we're looking for this sequence of interest or something related to it with an intron position at the R. Okay, so now we can take this sequence and just do a blast search of it. In this case, we're going to do a blast search of a protein sequence against the genome. So we actually need to get the genome. So we can just start by doing this with Medaka. No better reason than it's one of the three. So this is where the Medaka genome is, but remember that it's not in FASTA format, it's in GenBank format. So you can actually use a script called gb to fa.perl on the Medaka GenBank file, and that will create a Medaka FASTA file.